Welcome to Educational NishNova. Today we begin Class 11 Biology, Chapter 4, Animal Kingdom. In this chapter, we'll explore the fascinating diversity of animals, their classification, and the unique features that separate one group from another. Let's dive into the amazing world of animals. Chapter 4, Animal Kingdom. When you look around, you will observe different animals with different structures and forms. As over 1 million species of animals have been described till now, the need for classification becomes all the more important. The classification also helps in assigning a systematic position to newly described species. 4.1 Basis of classification. In spite of differences in structure and form of different animals, there are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, patterns of digestive, circulatory or reproductive systems. These features are used as the basis of animal classification, and some of them are discussed here. 4.1 1 Levels of organization. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. For example, in sponges, the cells are arranged as loose cell aggregates, i.e., they exhibit cellular level of organization. Some division of labor activities occur among the cells. In coelentrates, the arrangement of cells is more complex. Here the cells performing the same function are arranged into tissues, hence is called tissue level of organization. A still higher level of organization, i.e., organ level, is exhibited by members of Platyhelminthes and other higher phyla, where tissues are grouped together to form organs, each specialized for a particular function. In animals like annelids, arthropods, mollusks, echinoderms and chordates, organs have associated to form functional systems, each system concerned with a specific physiological function. This pattern is called organ system level of organization. Organ systems in different groups of animals exhibit various patterns of complexities. For example, the digestive system in Platyhelminthes has only a single opening to the outside of the body that serves as both mouth and anus, and is hence called incomplete. A complete digestive system has 2 openings, mouth and anus. Similarly, the circulatory system may be of 2 types. Open type, in which the blood is pumped out of the heart and the cells and tissues are directly bathed in it, and 2 closed type, in which the blood is circulated through a series of vessels of varying diameters, arteries, veins and capillaries. 4.12 Symmetry. Animals can be categorized on the basis of their symmetry. Sponges are mostly asymmetrical, i.e., any plane that passes through the center does not divide them into equal halves. When any plane passing through the central axis of the body divides the organism into 2 identical halves, it is called radial symmetry. Coelentrates, ctenophores and echinoderms have this kind of body plan. Figure 4.1a. Radial symmetry. Animals like annelids, arthropods, etc., where the body can be divided into identical left and right halves in only one plane, exhibit bilateral symmetry. Figure 4.1b. Bilateral symmetry. 4.1 3 Diploblastic and triploblastic organization. Animals in which the cells are arranged in 2 embryonic layers, an external ectoderm and an internal endoderm, are called diploblastic animals, e.g., coelentrates. An undifferentiated layer, mesoglia, is present in between the ectoderm and the endoderm. Figure 4.2. Mesoglia. Ectoderm, endoderm, mesoderm. Those animals in which the developing embryo has a 3rd germinal layer, mesoderm, in between the ectoderm and endoderm, are called triploblastic animals, Platyhelminthes to chordates. Figure 4.2b. 4.1 Forecoelom. Presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity, which is lined by mesoderm, is called coelom. Animals possessing coelom are called coelomates, e.g., annelids, mollusks, arthropods, echinoderms, hemichordates, and chordates. Figure 4.3a. In some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouches in between the ectoderm and endoderm. Such a body cavity is called pseudocoelom, and the animals possessing them are called pseudocoelomates, e.g., Aschelminthes. Figure 4.3b. The animals in which the body cavity is absent are called acoelomates. e.g. Platyhelminthes, figure 4.3. Syncosegmentation. In some animals, the body is externally and internally divided into segments with a serial repetition of at least some organs. For example, in earthworm, the body shows this pattern called metameric segmentation, and the phenomenon is known as metamerism. Notochord is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. Animals with notochord are called chordates, and those animals which do not form this structure are called non-chordates, e.g. porifera to echinoderms. 4.2 Classification of animals. The broad classification of Animalia, based on common fundamental features, as mentioned in the preceding sections, is given in figure 4.4. Echinodermata exhibits radial or bilateral symmetry depending on the stage. Figure 4.4, broad classification of kingdom Animalia based on common fundamental features. The important characteristic features of the different phyla are described. Phylum Porifera. Members of this phylum are commonly known as sponges. They are generally marine and mostly asymmetrical animals. Figure 4.5. These are primitive multicellular animals and have cellular level of organization. Sponges have a water transport or canal system. Water enters through minute pores, ostia, in the body wall into a central cavity, spongocoel, from where it goes out through the osculum. This pathway of water transport is helpful in food gathering, respiratory exchange, and removal of waste. Choanocytes or collar cells line the spongocoel and the canals. Digestion is intracellular. The body is supported by a skeleton made up of spicules or spongin fibers. Sexes are not separate, hermaphrodite, i.e., eggs and sperms are produced by the same individual. Sponges reproduce asexually by fragmentation and sexually by formation of gametes. Fertilization is internal and development is indirect, having a larval stage which is morphologically distinct from the adult. Examples: Sycon, Cypha, Spongilla, freshwater sponge, and Euspongia, bath sponge. Phylum, Coelenterata, Cnidaria. They are aquatic, mostly marine, sessile or free-swimming, radially symmetrical animals. Figure 4.6. Figure 4.6 examples of Coelenterata indicating outline of their body form. A. Aurelia, Medusa. B. Adamsia, polyp. The name Cnidaria is derived from the cyanidoblasts or cyanidocytes, which contain the stinging capsules or nematocysts present on the tentacles and the body. Cnidoblasts are used for anchorage, defense, and for the capture of prey. Figure 4.7 diagrammatic view of cnidoblast. Cnidarians exhibit tissue level of organization and are diploblastic. They have a central gastrovascular cavity with a single opening, mouth on hypostome. Digestion is extracellular and intracellular. Some of the cnidarians, e.g. corals, have a skeleton composed of calcium carbonate. Cnidarians exhibit two basic body forms called polyp and medusa. Figure 4.6. The former is a sessile and cylindrical form like hydra, adamsia, etc., whereas the latter is umbrella-shaped and free-swimming like Aurelia or jellyfish. Those cnidarians which exist in both forms exhibit alternation of generations, metagenesis, i.e., polyps produce medusae asexually and medusae form the polyps sexually, e.g. Obelia. Examples: Physalia, Portuguese man of war; Adamsia, sea anemone; Penatula, sea pen; Gorgonia, sea fan; and Meandrina, brain coral. Phylum Ctenophora. Sea ctenophores, commonly known as sea walnuts or comb jellies, are exclusively marine, radially symmetrical, diploblastic organisms with tissue level of organization. The body bears 8 external rows of ciliated comb plates which help in locomotion. Figure 4.8 example of Ctenophora pleurobrachia. Digestion is both extracellular and intracellular. Bioluminescence, the property of a living organism to emit light, is well marked in ctenophores. Sexes are not separate. Reproduction takes place only by sexual means. Fertilization is external with indirect development. Examples: Pleurobrachia and Ctenoplana. Phylum Platyhelminthes. They have dorso-ventrally flattened body, hence are called flatworms. Figure 4.9 examples of Platyhelminthes. A. Tapeworm. B. Liver fluke. These are mostly endoparasites found in animals including human beings. Flatworms are bilaterally symmetrical, triploblastic and acoelomate animals with organ level of organization. Hooks and suckers are present in the parasitic forms. Some of them absorb nutrients from the host directly through their body surface. Specialized cells called flame cells help in osmoregulation and excretion. Sexes are not separate. Fertilization is internal and development is through many larval stages. Some members like Planaria possess high regeneration capacity. Examples: Taenia, tapeworm, fasciola, liver fluke. That's all for this part. We'll continue this chapter in the next part. Stay tuned with Educational NishNova and don't forget to like, share and subscribe.